Hi, so today I'll be expanding on the topic of trehalose, which I started talking about in January, so let's get started. Alright, so today we'll be entering the realm of supramolecular chemistry, which is basically how we transport different molecules that can benefit the body around the body, using delivery systems that are also biomolecular. So one of the most common delivery systems is by the use of graphene, carbon nanotubes, or fullerene, which are just different forms of polycyclic carbon atoms. So, what are polycyclic carbon atoms? Basically, carbon atoms which are formed in a sort of hexagonal pattern, sort of like in this ring up here, with alternating double bonds. Or when we're talking about hybridized orbitals later, they'll be hybridized orbitals alternating with regular sigma bond. So let's start at the beginning. We want to transport trehalose around the body for therapeutic purposes, but how do we do it? Our first thought is to ingest it, which is probably the easiest way to do it, but the problem with that is you have to ingest a very high dosage of trehalose because in the digestive system, an enzyme called trehalase breaks down trehalose into components that cannot be used by the body, which is not what we want. Another way to do it is biopsy, which is getting it injected into yourself. Injections have a lot of hassle, and we do not want that hassle. So, how do we ingest it and get it around the body? Well, our first thought is to make a sort of molecular cage for it. And one of the most common molecular cages is fullerene, which is the carbon structure we discussed in a spherical shape. So, fullerene is very stable and a great supercapacitor. An endohedral fullerene isn't a supercapacitor, so it won't conduct electric currents when it's going around the body. So what's the problem with using endofullerene? All right, so looking up here, endofullerene, one ligands attached to it. Ligands are just any molecular components that kind of attach around a central compound. When the ligand's attached to it, it'll collapse, and then the molecule that is within it won't be able to get out. Also, making a fullerene big enough to transport trehalose is very difficult, or to be more specific, very costly. Also, very difficult to open a fullerene cage because they are so stable once they collapse into a sort of supersymmetry. So, the most optimal solution here is a carbon nanotube, as I drew here, which kind of progresses as a tube in a one-dimensional direction. So a carbon nanotube is made up of that carbon structure that I discussed earlier, and it has quite a few components to it in order to transport the trehalose. By the way, the trehalose is these two structures. So the focus of this is not therapeutics anymore. We want to target α-Synuclein, a protein aggregate that causes many diseases in the brain, such as Huntington's and heart disease. So this magnet and this magnet, they are going to represent the α-Synuclein protein aggregates in this diagram. So we have these protein aggregates, but how are proteins formed? Let's move down here for a second. So in a cell, in a cancerous cell to be specific, because this cell must have cancer to produce such a protein aggregate, it's uncontrolled cell growth. So the DNA, it's double stranded first of all in a helical structure. It has a 3' end. Each strand has a 3' end and a 5' end. They have opposite ends on each side. And so what happens is that these are translated to a messenger RNA code, which is showed right here, and it's single stranded here. And then these are transcribed into a protein at a ribosome. So this protein, this cancerous protein, because this is a cancer cell we're talking about, is now the α-Synuclein aggregate. So we have to stop this process somewhere in the middle in order to get rid of this α-Synuclein or prevent it from forming. So how do we do that? Well, let's look at the diagram right here. So the first thing we've got is plasmids. We'll actually discuss that a bit later. So let's move to the polyethylene glycol. Polyethylene glycol is really good for holding together this whole chain, this branched polyethylene glycol, which in the end is what helps us deliver the trehalose by attaching it to the carbon nanotube. The peptide is this zigzaggy line right here, and the peptide is really what attaches the trehalose to the carbon nanotube. So then we have the trehalose, and we have RNA degrading material. Now, what is this RNA degrading material? It's attached by a disulfide bond. It's usually something called siRNA, which just stands for something that will get rid of RNA. And there are many molecules that do this, so we can pick any one of them. We'll just call this siRNA for now, attached to the molecule by a disulfide bond, as seen here by the two S's. So we have siRNA. So what does siRNA do to stop this cycle? Well, it can do one of two things, or both of them. It can excise the exons, or it can directly degrade RNA, and it can kind of do a combination of both. But let's just talk about each one of them individually. So there's three classes of really RNA degrading techniques. So one of the classes is hydrolyzing the 5' end or degrading the 3' end. Those are two of the classes. And this is just degrading each end. That characterizes aging in the strand, and it also just kind of stops the process, prevents the alpha-synuclein protein from forming. Also, we could go into the nucleus, which is the difference between exonucleases and endonucleases. We can go into it and cut it internally, which is what it means by excising the exons. So there are really two parts of a strand. There are really two parts of a strand of this messenger RNA strand. And so these two parts are the introns and the exons. And then you have the 5' and 3' ends. Introns are cut out, and the exons are the parts that are actually displayed in the gene pool. So if we cut out the exons, then the gene will be prevented from being displayed, and therefore the alpha-synuclein will never be formed. Alright, so now let's talk about the plasmids. Coming back up here to the plasmids. What are plasmids, first of all? Plasmids are little portions of a bacteria where non-centralized DNA is found. So it's not like the nucleoid of the bacterium where there's a whole bunch of DNA found, but it is sort of decentralized. It is just lying all around the place. And it helps the bacteria conjugate. And conjugation is just a bacteria's way of sexually reproducing, which helps it fight against antibiotics and just kind of work against environmental factors. And so that's why sexual reproduction in general is just a benefit. It provides variety so that a population can survive with a variation of genes. So what if we took these plasmids, which are now our preferred strands of DNA, and put them in where we cut off the RNA to make the cell work normally again? That would be the optimal solution so that the cell doesn't die out. Instead, it starts working normally again. So we have our plasmids attached to it. Now, what is this fifth one right here? This fifth one is a pi-stacked aromatic drug molecule. So what does it mean when it says pi-stacked? Alright, going into some chemistry right here. Hopefully you remember this from your chemistry class. There are the two most basic types of orbitals are s orbitals and p orbitals. An s orbital looks like a sphere, and a p orbital looks like a sort of figure eight shape. And these are three-dimensional orbitals with an x, y, and z axis. These are just because the lowest possible energy a wave can have, a standing wave can have, is like that. And if we rotate that, we get a sphere. And the second lowest possible energy that a wave can have is if it has one node. That means it intersects this once. Now if we make this three-dimensional, then it becomes a p orbital. And it goes on and on after that, having 2 nodes, 3 nodes, or even 4 nodes. And so these are 1 and 2 node orbitals. So let's go back to the carbon structure for a second. So there are single bonds and double bonds that are alternating. But really, instead of being a shape sort of like this, where they're alternating like that, it's more of a circle where the electrons are going around. This is called a polycyclic compound. Why do we have a pi-stacked aromatic drug in the background? And really what this is, it's for carrying cancer preventing cells. So doxorubicin is a cancer treating molecule which can be carried. In pi stacked, what it really means is, so sigma bond happens when two hybridized orbitals, which means hybrid is any two things that are combined, right? So hybridized orbital is one where the s orbital and the p orbital are combined. So if you have an s orbital like this, and a p orbital like s orbital like this, and a p orbital like this, if you combine them, then you get something like that. So that's basically what a hybridized orbital is. So if you have two hybridized orbitals facing each other, and they overlap, that's a sigma bond. And that is the basis for single bonds. Now how do double and triple bonds form? This happens when you have two p orbitals or higher than p orbitals that form when they overlap one another parallel. And this is just one pi bond. It's not two. This parallel structure creates one pi bond. And so when you have this, it creates a stable structure which is capable of carrying molecules. And so this is why we have a pi stacked aromatic drug molecule. So one thing that we want to know is, how do we open this molecular cage sort of device? Or how do we break these bonds so that the trehalose can be released into the cell? Well, the answer here is we can either change the pH, which is a common technique used to open molecular cages, or we can shine a certain range of the electromagnetic spectrum onto this sort of structure, and this will loosen the bonds up between the trehalose and the structure, depending on the wavelength that you shine on it. And then that would release the trehalose, and it would be able to work with the α-Synuclein. And that is basically how this whole trehalose delivery process works.